Endowed Chair in Cancer Research and the Associate Director for Basic Research of the University of Colorado Cancer Center. So she received her undergraduate degree in biology with a minor in chemistry from the State University of New York, and her master and Ph.D. degrees in biochemistry from the University of Rochester. And then after completing her postdoctoral fellowship under the mentorship of author Ph.D. at the Dana-Farber Cancer Institute and Harvard Medical School, Dr. Ford joined the University of Colorado School of Medicine in 2001. Since then, she has been running a funded laboratory that focuses on the role of developmental regulators, particularly 6-1 and IA transcriptional complex in breast cancer and pediatric tumor genesis and metastasis. Her work actually spans a very diverse area, and you'll hear some of that, from understanding the parallels between normal development and tumor genesis and basic molecular pathways that contributes to metastasis, including EMT, to more translational aspects of developing novel therapeutics for cancer therapy. So to date, she has published more than 80 peer-reviewed articles and some reviews, and these include lots of the prestigious journals such as Cell Reports, Oncogene, Cancer Research, Nature Communications, and she has also mentored numerous undergraduate students, graduate students, and postdocs. And many of I would say most of the alumni actually stay in science field. Some of them goes to a school, university as a faculty, and in different countries, in the United States, in China, in Brazil, and also some go to industry such as in Amgen or Johnson & Johnson and stuff like places like that. And she's actually also the director of a biomedical science graduate program for 8 years in the past. She also received numerous awards. I guess I don't have to need to go through all of them. I think this explains the very interesting work that she works on and also the very diverse field she covers. And let's listen to her talk. The title is Understanding Tumor Heterogeneity and Cellular Plasticity as key drivers for metastasis.
Thank you. So can everyone hear me okay? Yes. So I wanted to thank Shui for that introduction. That was really, really kind, and I'm excited to be here. It's great to be in person. I've just started giving talks in person again, and it's so nice. So I'm going to keep my mask off. I hope that's okay up here, just because it's easier to talk and for you to hear me that way. So can everyone, so on my computer, it flicked to a different mode, but I think it's okay. You see it okay, and everybody who's online sees it okay? Like, you don't see presenter mode, you just see the slide? Yes? Okay, good, because here it went off of that. I don't know. Anyway, it's fine. Okay, great. So I'm going to talk today about some of the work we're doing in the lab around tumor heterogeneity and cellular plasticity. And just to give you a little idea of what we're interested in the lab is that we're really interested in the parallels between normal development and tumor genesis. And particularly, we're interested in temporal and spatial gene regulation that occurs developmentally, and how genes that regulate things like self-renewal, growth, survival, motility, and invasion get re-expressed in tumors, but now are not temporally and spatially regulated in the same way, so are acting out of context, wrong place, wrong time, to stimulate tumor progression. And the reason that we're so interested in those parallels is that many of these genes that we work on that are transcription factors that are highly expressed and important in early embryogenesis are actually down-regulated in differentiated cells. And so if cancer is a real problem to target because it's a disease of self. So if you can find genes that are not expressed in most adult normal cells and they get re-expressed in cancers, they may be potential ways or therapeutic targets. And so that's something we're very interested in. So as Shuji mentioned, I'm really interested in the epithelial to mesenchymal transition and how that becomes reactivated in cancers. And I'm sure you've heard from her work a lot about neural crest epithelial to mesenchymal transition. So I probably don't have to introduce this too much, but this is a process in which cells that begin epithelial undergo a transition to a mesenchymal state. And in cancers, and developmentally, this is very important for numerous different tissues, including neural crest development. But in cancer, what's actually thought to happen, particularly in epithelial tumors, so I'll talk mostly about breast cancers, which are epithelial tumors, it's thought that a subset of cells in the tumor will undergo a very similar process as to what happens developmentally, and that's an EMT, and that those cells then become more migratory and invasive, and it's those cells that can move from the primary tumor to secondary sites. And my lab is really interested in that process of metastasis, so the movement of these cells from the primary tumor to distant sites, because really in most tumor types, not all, but in most tumor types, what kills patients is metastasis of those cells to critical organs and not where they start dividing in the primary tumor in the first place. All right, so how is this epithelial to mesenchymal transition thought to play a role in metastasis? So what's thought to happen, again, is if you're in a primary tumor that's of epithelial origin, a subset of these cells will undergo an EMT, and those cells are thought to be more migratory but also more invasive. So they secrete enzymes like matrix metalloproteinases that can digest membranes, and that enables them to get into the bloodstream, to go through the circulation, and then when they exit at a secondary site, what's been thought, based on a lot of experimental data, to occur is that these cells will then get out of the bloodstream and they'll undergo a reverse mesenchymal to epithelial transition and then grow out at the secondary site. And the reason that this reverse process is thought to occur, which again has been shown to occur experimentally, is because when you look at metastases at secondary sites of epithelial tumors, they look epithelial. So they look like the cells at the primary site, so they don't really show that they've undergone an EMT. But what has been shown very clearly is that, and this is actually, there's correlative human data too, is that during the process of chemotherapeutic treatment, when patients come in and they have a cancer, that actually the epithelial tumor cells tend to die out, and that these mesenchymal tumor cells survive, and then you will see a more mesenchymal subtype at the secondary site. All right, however, a few years ago, some fate mapping experiments in which they essentially tagged the cells that had undergone an EMT in the primary tumors actually argued that MET doesn't happen. And in fact, that the cells that undergo an EMT maybe are sort of passively allowing the other epithelial tumor cells to get into the bloodstream, but that they never undergo this reversion. They did show that it was important for chemo resistance, but not for just general metastasis. And to us, this was pretty surprising because there are thousands and thousands of papers and experimental models, et cetera, that show that an EMT can actually increase metastatic dissemination. And so one of the things we thought about, we started studying at that time, was whether or not the cells that undergo an EMT in a primary epithelial tumor can crosstalk with other cells in the tumor such that they change their characteristics and actually actively make them more migratory and invasive without making them undergo a clear EMT. So if that happens, when you do fate mapping, you wouldn't see the effects of EMT on metastasis. And so this is something that was actually inspired by some earlier studies, but that we wanted to then more carefully dissect and see if it actually really happens. And so I had two really, this is showing up in a really strange way, but I had two really talented graduate students in the lab who partnered to try to look at whether this can occur. And so what we started out with was just very simple experiments where we took human mammary epithelial cells that are RAS transformed, these are from the Weinberg lab and were given to us, that either express GFP, so these are known to be tumorigenic but non-metastatic and have not undergone an EMT, or they express two main transcription factors known to play a role in EMT, either snail or twist. These cells are known to have undergone an EMT, and they're also more metastatic in vivo. So what we did was just simple scratch assays, and somehow this isn't showing up very well, but what you can hopefully appreciate is that the cells that have undergone an EMT are much more migratory after 5 hours than those cells that have not, which really don't move at all. But if you mix these cells, and we mix them at a ratio of 1 to 10, so we took 1/10 as many cells that had undergone an EMT and mixed them with cells that were epithelial, what you find is that the green cells, so those are the epithelial tumor cells, actually start migrating at a much higher rate in the presence of these cells that had undergone an EMT. And in fact, when we quantitated that, this is quantitation only of the migration of the green cells. What you hopefully can appreciate is that they're migrating much more effectively in the presence of 1/10 as many cells that have undergone an EMT, which may represent what happens in an actual human tumor. And in fact, those cells that are migrating and in this case, migrating more, they have no change in their EMT markers. So if you check the typical markers that you may have heard of from Shui, Shuyi, sorry, I don't think I pronounced that correctly. And you were so good about pronouncing my name. So if you check those markers like E-cadherin and N-cadherin, et cetera, they don't change their surface markers, yet they're migrating and invading much more efficiently. Okay, so how is this happening? So we tried to figure out molecularly how the cells were talking to the other epithelial cells in the tumor. And one of the things we wanted to understand was this molecule that we work with quite a bit called 6-1. It's a homeo-containing transcription factor, so it's a critical developmental regulator. It's known to be important in the development of numerous tissues, including in epithelial to mesenchymal transitions that occur during muscle development. We wanted to know if this transcription factor, which had been shown to be in sort of this complex web with twist and snail, was actually playing a role in this process. So again, we're interested in this particular transcription factor because it's, ooh, what just happened? Just timed me out or something. Sorry. Is it back? Okay. So we're interested in this particular transcription factor because, again, it's one of these genes that's expressed in early embryogenesis but downregulated in most developed tissues. And so we wanted to know whether this is something involved and if there's a way to target it. And again, we know it promotes an EMT both in development and cancer, and that previous studies from our lab and others had shown that it promotes metastasis in a number of different cancer models. Okay, so the first thing we asked just was a very simple question. Is 6-1 regulated downstream of snail and twist? And sure enough, it is. So if you overexpress snail or twist and make these cells undergo an EMT, you see you have a dramatic increase in 6-1. And of course, if you knock that down, you lose it. And what's the net effect of that? So I'm sort of going through this briefly because I want to talk about some newer data, and this is published. But essentially what we had shown is that if you take media off the cells that have undergone an EMT and transfer it onto those cells that are epithelial, and that's this experiment here. So here's the media off control cells. These guys, sorry, off the cells themselves. These migrate very well. If you then take media off control cells, put it on control cells, they don't migrate. If you take the control cells, so by control I mean the epithelial counterparts, and add media from the cells expressing snail, now you see you increase their migration. And if you take the media off the snail-expressing cells or the twist-expressing cells in which you've knocked down 6-1, so you've lost that transcription factor, and transfer that, now you don't increase the migration. So there's something that twist and snail are regulating 6-1, and that's regulating something that gets into the media of these cells that talks to the other epithelial tumor cells. Oops, sorry about that. And the same is true for invasion. We can do this for a number of different characteristics. And again, no change in EMT markers with those cells when you transfer the media. Okay, so that shows that 6-1 is necessary for this phenotype. Is it sufficient? And so what we did is we overexpressed 6-1 in a breast cancer cell line that's very epithelial, so it's not undergone an EMT. And what you see here is when we do that, this is actually this cell, you can see the change in some EMT markers. In this particular model, when you take media off cells that express 6-1, what happens is those cells gain more aggressive phenotypes, but they actually also do change EMT markers. So it's not a one-size-fits-all. The context matters. But what I will say, and I can maybe talk about this later because I think we understand somewhat why this happens, I will say that we know that 6-1 is sufficient to cause paracrine-mediated changes in the epithelial breast tumor cells. So again, if we look down here and we take media off of cells expressing 6-1, what you see here is now we downregulate E-cadherin, a known mesenchymal marker. We also can increase the resistance of these cells to anoecus. So that's cell death when they're not attached to a matrix, right? And so in all cases, there's something secreted by the EMT cells that affects the epithelial tumor cells. In some cases, depending on the model, it makes them more migratory and invasive without changing the markers. In other cases, it actually does induce what I would call more of a hybrid or a partial EMT based on our characterizations. Okay, so when we did this work, a lot of people complained that we weren't doing this. We were using very artificial models, right? We were either overexpressing the transcription factor 6 or we are overexpressing twist or snail, but we were always putting a transcription factor in there to induce EMT. So we wanted to see if the same thing would happen if we used spontaneous models of EMT and epithelial cells. So what we chose is two models that were derived from MMTV, polyoma middle T tumors. These are mammary tumors that arise in mice when they express the polyoma middle T. This one line was called MET1. It's highly motile and highly metastatic. And this other one it has low motility and low metastatic. So the first thing we did was just characterize them with respect to EMT. So do they follow the paradigms? And what we saw when we checked a number of markers associated with EMT is that the non-metastatic lines had high E-cadherin expression compared to the metastatic. And they had the reverse. They had high N-cadherin and vimentin, showing just in a broad way that the highly metastatic lines have more EMT features than the non-metastatic lines. So now we figured, okay, we haven't manipulated these. These are naturally occurring in response to polyoma middle T. These are naturally occurring tumors in these mice. So are they dependent on 6-1? So similarly, wow, this is moving ahead on me. Similarly, we saw that the MET1, so these are the highly metastatic lines, have high 6-1. Why is it doing that? And that if you do the same types of experiments, so you take the conditioned media off of the MET1 cells and put it on the DB7 cells, you make them more migratory and more invasive. And if you knock down 6-1 in those cells before collecting the conditioned media, you actually cannot induce migration and invasion to the same degree. So this is another system in which you could affect migration and invasion, crosstalk, by using the media of one cell type and putting it on the other. But there's no change in the EMT markers in the DB7 cells that are treated with the conditioned media from the MET1 cells, despite the alterations in migration and invasion. And the reason I bring this up again is because fate mapping would miss all this crosstalk. Okay, so molecularly, how is this all happening? So we did a lot of experiments in which we took conditioned media off these cells, and we did fractionation and biochemical experiments to try to figure out what it was in the conditioned media that was actually causing this crosstalk, and couldn't figure it out. And then this is why multidisciplinary science is so important as a great example of this, because I went to a seminar where somebody was working on Drosophila eye development, nothing to do with what I work on, but they happened to mention that Cynoculus, which is the Drosophila homologue of 6-1, regulates Hedgehog in the context of fly eye development. And that made me really curious, because Hedgehog has been implicated in cancers in some of these processes that we're looking at, migration, invasion, et cetera. And so I went back and I said, well, okay, is Hedgehog signaling activated when you take the conditioned media off these EMT cells and put it on our epithelial tumor cells? And what I did first to look at that was I took a reporter that has a 7-Gli binding site. Sorry, so I should back up for a minute. When Hedgehog binds to its, why does that keep happening? How did I get it back last time? Yeah. Okay, so when Hedgehog binds to its ligand, it activates a signaling cascade that ultimately results in a transcription factor called Gli1 being activated and binding to and turning on the expression of a number of genes. So the first thing we did was, now it doesn't want to move forward. Odd. Will that do it? Okay, I'll just say, okay. So the first thing we did was just take a reporter that we got from a collaborator who's at Baylor College of Medicine, Mike Lewis, that had 7 Gli1 binding sites in the DNA that we could then had GFP downstream, so we could see if we were activating Gli-mediated transcription in all these models when we transferred the media. And what you can see pretty clearly is that in the cells overexpressing 6-1, the MCF7 cells, you transfer that media onto the control cells, you activate Gli. In the snail model, you can activate this reporter with the media from the EMT cells, the snail cells. That gets removed when you knock down 6-1 in those cells. And similarly, in this DB7 MET1 system, same thing. You can activate the reporter. It gets removed if you get rid of 6-1. Interestingly, however, Hedgehog ligands were only activated in a 6-1 dependent manner in one of these three systems, and it was the system where you could see the EMT markers change. So we actually think that Hedgehog is important for that EMT marker change, but not important for activating this pathway in all the contexts. And so we wanted to understand what actually activates the pathway then. It's not the Hedgehog ligand. All right, so if you go back to this, there are numerous inhibitors that you can use in order to inactivate the pathway at different points. This inhibitor actually inactivates smoothen, one of the key receptors required for the signaling. This cyclopamine, this inhibits Hedgehog, the ligand itself. Cyclopamine inhibits smoothen, which is a receptor required for the signaling. And then there's actually an inhibitor called GANT that specifically inactivates the transcription factor. So I'm just going to show you the GANT data in all contexts. Again, these are conditioned media transfer experiments, all on the control cells, so meaning the epithelial tumor cells. So we have epithelial tumor cells. If we treat them in their own media, they don't migrate well. Treat them in the snail media, they migrate efficiently. If we take the snail media, but we add in GANT61, so to inhibit glee activation, you can see we remove that. And the same is true in all these contexts. And also in the 6-1 system, the MCF761 system, we can also show that if we use GANT61, we can restore epithelial E-cadherin, making these more epithelial. So what this shows you is that the activation of glee is actually responsible for these aggressive phenotypes in these cells. However, cyclopamine, which remember is the smoothened inhibitor, it only inhibits the aggressive phenotypes in the system where 6-1 is expressed on its own, so where Hedgehog is actually affected. So what that tells us is that since GANT, remember, is the inhibitor of glee, what that tells us is that they're all acting through glee, but not via the canonical smoothened and Hedgehog signaling pathway. So that still left us not knowing how this happens. And I'll just run through this quickly. Same phenotypes are seen in this DB7-MET1 system. Again, if you inhibit glee, you inhibit those aggressive phenotypes that are transferred when you use the conditioned media off of EMT cells. Okay, so does this matter at all in vivo? So one of the things we wanted to know is whether this crosstalk is actually happening in the context of animals in vivo. And so what we did here is we did cell mixtures where we luciferase tagged the epithelial tumor cells, and we RFP tagged the cells that had undergone an EMT. In this case, we used the 6-1 model, but we've used numerous models. This loves to move on its own. Sorry, let's back up. And what you can see is here now we could track the metastasis of only the luciferase tag cells, so the epithelial tumor cells. And what you can hopefully appreciate is when you mix them with the EMT cells, they metastasize much more efficiently, right? Importantly, if we then treat these animals with GANT61, so that's the glee inhibitor, we can inhibit the metastasis of these tumors pretty dramatically. And sorry, this is inhibiting metastasis of the luciferase cells, so the epithelial tumor cells. It also affects metastasis of the RFP, so the EMT cells, but to a lesser degree, actually. So that's pretty striking. The metastasis was really inhibited. And it's important because of this. So clinically, cyclopamine derivatives, which remember, this is upstream in the pathway, have been used in clinical trials in tumors like basal cell carcinomas and medulloblastomas, where there are known mutations in the signaling pathway that constitutively activate Hedgehog signaling. However, these same inhibitors have been really ineffective in solid tumors like breast cancers, even though we know Hedgehog signaling is active in those tumors. So you can look at glee transcription and you know it's being activated. And so what we would argue is if you've got a heterogeneous tumor where a subset of cells have undergone an EMT, those may be able to signal through this pathway via Hedgehog, but they can also signal via upregulating, and I didn't tell you this yet. I'll tell you this in a minute. But we know what the secreted factor is. And by upregulating glee in a non-canonical way, they can actually, if you use the inhibitor, to cyclopamine, it wouldn't work, right? Because you have another way to activate this pathway. However, if you used an inhibitor that was more downstream, like glee, theoretically it should work. So is this the case? So we coupled with Mike Lewis again at Baylor College of Medicine, and we used patient-derived xenograft models and tested them either with a smoothen inhibitor, so that would be upstream in the pathway, or with the glee inhibitor that's downstream. And what we can see here is that if you use the glee inhibitor, you dramatically, this is tumor doubling in this case is what we were measuring, but you dramatically decrease that, whereas if you use a smoothen inhibitor, you have almost no effects. So what this suggested to us is that the crosstalk pathway might actually be important in this context. Okay. So I sort of gave it away. I obviously had my slides a little out of order. But what is this secreted factor? So it took us a long time to figure this one out too. But it turned out that in all contexts, in all models, one of the things that we saw that was secreted when you overexpress 6-1 or in a 6-1 dependent way. So it was upregulated when you added in snail, when you overexpress snail. But if you knock down 6-1, you lose it. And that's VEGFC. And this is just an ELISA to look at that in one of our model systems. So this is interesting because VEGFC is not canonically an activator of glee signaling. So VEGFC canonically is actually important. I don't know if any of you follow this at all, but it's important in lymphangiogenesis and the growth of lymphatic endothelial cells. But in this context, what we could show is that indeed it could activate glee signaling. So again, if you take conditioned media from 6-1 expressing cells, you can see activation of that glee reporter. If you take an antibody or an siRNA against VEGFC, you can diminish that. And I'm not going to walk through it in each model, but it happens in all the models we looked at. VEGFC is secreted, and it actively activates glee in those epithelial tumor cells when you put the media on it with VEGFC in it. And we know that VEGFC is important for the aggressive phenotypes. So, for example, if you look at the MCF7 cells and we use E-cadherin as one of our readouts, so cell surface E-cadherin, we can show that if you add the MCF7-6-1 conditioned media, you lose that E-cadherin surface expression. If you neutralize VEGFC with an antibody, you can restore it. And similarly, looking at invasion in our other model systems, if you use a VEGFC antibody, you can inhibit the ability of the EMT cell media to cause increased invasion of the epithelial tumor cells. So we knew this was really important for these aggressive phenotypes. Okay, so we know it works this way. So snail or other EMT transcription factors are activating 6-1, which then leads to increased expression of VEGFC, which ultimately results in increased secretion. That acts on particular receptors. We worked this out. Daguan Kong, a visiting student in my lab, actually did all this molecular pathway analysis. What it does is it works through this combined receptor of VEGFR3 and neuropilin-2, and that activates glee in a non-canonical way. So again, we wanted to go back to the in vivo experiment and prove that it's actually the crosstalk between the two cell types, the epithelial tumor cells and the EMT tumor cells, that matters for this metastasis of those epithelial tumor cells. And so this is a little complicated to look at because there's a lot of lines, but suffice it to say we did another mixing experiment. This time we used a different model, so it's the DB7-MET1 model, where we labeled the cells either with luciferase if they were epithelial or with RFP if they were, actually in this case I think we might have done unlabeled if they were EMT, had undergone an EMT. And then we either knocked down VEGFC in the cells that had undergone an EMT, or we knocked down neuropilin-2 in the epithelial cells. And we asked, are those required to mediate metastasis of the epithelial tumor cells? And so what you can first appreciate is that just growth of the tumors is dramatically affected by mixtures. So if you have the DB7 cells, those are the non-EMT, so they're the epithelial cells growing with themselves. They don't grow very efficiently. If you have them growing in the presence of these EMT cells, they grow much better. And then if you do any combination of knockdowns, either VEGFC, knockdown in the EMT cells, or neuropilin-2, the receiving receptor in the epithelial cells, you decrease that increased growth. And in fact, if you look at survival, this is looking at luciferase signal at the distant site. So this is looking at metastasis. This is primary tumor growth. If you measure survival, again, this is a little hard to look at, but just focus in on the red line. If you mix the DB7 and MET1 cells, these mice, don't do well at all. And then if we go here, look at this line. If you take your epithelial tumor cells with neuropilin-2, the receptor knocked down, and you add in your mixture of your EMT cells that still have VEGFC, you can see that the survival is much better. So in all cases, survival is better if you don't have either receptor on the epithelial tumor cells or the VEGFC being made by the EMT cells, suggesting that, again, this crosstalk is actually important in vivo and is one way in which EMT contributes to metastasis. Okay. And so these data argue that inhibiting this whole axis is going to reduce both primary tumor size and metastasis of the epithelial tumor cells. And so this is something that we're really actively pursuing in PDX models by trying to use different components of inhibition of this pathway to see what we can do to this metastatic dissemination. I don't see time. How am I doing on time? Just so I know how to... Okay, great. All right. So one of the questions we had then is I just told you that you can inhibit glee downstream. You can inhibit these guys, and there are drugs and antibodies that will inhibit VEGFC or VEGFR3, for example, or neuropilin-2 even. But the thing is that if you did that, you would inhibit the crosstalk, and therefore you might inhibit the metastasis of the epithelial tumor cells. But one of the questions we've had for a long time is, what if you went further upstream? Because if you did, this would inhibit EMT in the tumor cells themselves, and it would inhibit the crosstalk, and therefore that whole chemoresistance phenotype that you see, which is really dependent on EMT in the tumor cells themselves, you might remove that too. And so we've been really interested in figuring out ways that we could actually drug 6-1. So this isn't easy because it's a transcription factor. And transcription factors are notoriously difficult to drug. And so one of the ways that we've been going at this is to go for its partner called eyes absent. And eyes absent is actually a critical partner for 6-1 to mediate transcription. 6-1 doesn't have any activation or repression domains, so it really just serves as the binding sequence to which the Aya comes in and then acts as the transcriptional co-activator. So this is kind of how the complex looks. And what's interesting is that, like 6s, Ayas are expressed early in development. They're involved in all the same processes that the 6s are, and in fact, they're known to act together developmentally. And even in human diseases, they're known to act together. So we know in the human they act together. For example, there's a syndrome called branchiotorenal syndrome, where these patients have kidney malformations, craniofacial defects, hearing loss. They are found to have mutations either in 6s or Ayas. So they're known to act together in human development too. And they're known to be altered in the same cancers. So to us, this was a great way that we could maybe try to inhibit this complex. And the reason is as follows. We could do two things. We could try to inhibit this interaction, but also it turns out that Aya is not only a transcriptional activator, but it's a phosphatase. And so it has enzymatic activity. And while phosphatases are not as easy to target as kinases, they are still targetable. So this is something that we thought we could maybe go after. And so the first thing we wanted to do is look at was, are Ayas important for EMT and breast cancer? Do they work with 6s? And so I'm not going to go through all this data, but what I can tell you is, dependent on the context, so this first paper we published was showing that Aya2 is required to mediate the pro-metastatic functions of 6-1. This was in a luminal breast cancer model, and this was in a triple-negative breast cancer model. I'm not sure if everyone knows the distinction, but they're different subtypes of breast cancer, and they're very different. And what we've found is that dependent on the Aya, or dependent on the type of cancer, a different Aya may play a role, and they may compensate for each other. So multiple Ayas are important. And this is just some, again, marker analysis to show you that when you knock down Aya3, because this is unpublished, you knock down or you lose a lot of mesenchymal markers in these cells. All right. What's interesting is when we did some of these studies, in the context of the triple-negative breast cancers, we were working with immune-competent models. And when we would stain these models, we would notice that, so this is a breast cancer that's actually highly mesenchymal. You can see it's got high vimented expression. When we knocked down Aya3, which was the predominant or the dominant Aya in that setting, you can start to see E-cadherin re-expressed, so you're partially converting. You're not, there's still a ton of vimentin, right? But one of the things we noticed right away is huge infiltration of immune populations. So the immune microenvironment was very different in the presence or absence of Aya3. And in fact, I'm not going to go through all this data because we published this a couple of years ago, but essentially what we showed is that when we knocked down Aya3, we dramatically affected primary tumor formation, but that it wasn't dependent on proliferation. We didn't see massive differences in proliferation, but instead on the amount of CD8 positive T cells that were infiltrated into the tumor. So when you knock down Aya3, you had a dramatic increase in CD8 positive T cells, which are your cytotoxic T cells. And actually, so this is the growth of these tumors. This is wild type. This is Aya3 knockdown. And if we depleted the CD8 positive T cells, which are these dotted lines, you could pretty much restore a lot of the growth. And what that suggested is that while they're causing an EMT, one of the dominant functions in this context for Ayas is actually affecting the immune microenvironment such that it allows these tumors to evade the immune system. And we actually spent a lot of time working out molecularly how this happens, and I'm going to get back to this, so this is part of why I wanted to tell you this. But we know that Aya3 regulates MYC, and I'm going to tell you it regulates it on several levels, and that actually regulates a molecule called PD-L1. which when it's expressed on the surface of cells, it causes T cell exhaustion, CD8 positive T cell exhaustion, and therefore decreases the function of the CD8 positive T cells against the tumor. And again, we published this in a couple back-to-back studies that were sort of, this one showed more of the immune biology, and this showed more of the molecular context as to how it happened. Okay, and observed in multiple models. What we really wanted to do, though, again, my lab is really interested in metastasis, and that was all primary tumor growth. And so I had another graduate student who came in, Connor Hughes, who wanted to understand if it was also affecting metastasis similar to 6-1 in this model. So he did the same knockdown experiment and then looked at tumor growth, and this is a murine mammary carcinoma model. And you can see, again, huge differences in primary tumor growth. So, of course, when he measured metastasis, so this is metastasis here, it was hugely different. But that's a really hard thing to assess, whether that's due to the size of the tumor versus metastasis per se, because the tumors are so different in size, right? And so, in fact, of course, survival is much better if you knock down IA3. So what he did was he actually went back and did the experiment again, and then he removed the tumors at the same size, and he asked the question as to whether then, once the tumors were removed and they weren't allowed to get different sizes, did the metastasis, was it differentially affected? And what he saw is still pretty large effects on metastasis, suggesting that IA3 does more than just affect primary tumor growth, but it might also affect the metastasis of these cells. And then when we looked more carefully at the tumors, in addition to changes in CD8 positive T cells in these tumors with IA3 loss, there were massive changes in a number of different cell types that are involved in the innate immune response to tumors. And I'm not going to go through all of these, but suffice it to say that there were just big immune differences. And so he thought that probably the MYC PD-L1 axis couldn't explain all of this. That might be a portion of it, but not all of it. So he went back and he did RNA-seq on our IA3 knockdown cells. And one of the top pathways that came out that was actually very correlative with the relative amount of knockdown, because this is a much better knockdown than this one, was changes in the inflammatory response, and in particular in NF-kappa B signaling. And so, in fact, when he looked at the genes that were regulated by IA3 knockdown and then looked at what sort of transcription factors regulate those genes, what he found is that a key regulator of NF-kappa B called REL-A, it's part of a component of NF-kappa B, is regulating a lot of these genes that are changed with IA3 loss, suggesting to us that IA3 is not only playing a role in MYC signaling, but also in NF-kappa B signaling. And this just shows you, I'm going to real quickly show you some of the data. We've got a lot of data around this now. But what he could show is that actually IA seems to be affecting nuclear localization of REL-A. So if you knock down IA, you can see REL-A is pretty present in the cytoplasm, but you see that there's significantly less REL-A in the nucleus, which is required for it to activate transcription. So it's somehow affecting the localization of REL-A. And that, of course, leads to massive alterations in NF-kappa B activity. So this is a luciferase assay that shows that when you knock down IA, you have significantly less NF-kappa B. So this is super interesting to us because, remember, we have these massive immune changes. And NF-kappa B signaling is known to regulate a lot of cytokines. It's important for cytokine expression. Before I get to that, what I will say is that we've also done analysis in human data sets. So these are human tumors from the Metabrit data set. It's a very large breast cancer data set. And basically just looked at which genes are co-expressed with IA3. And then once we got those gene lists, we actually put it through GSEA analysis. And what you can hopefully appreciate here is that the pathways that come up are the pathways that we've been looking at. MYC, TNF-alpha, inflammatory responses, NF-kappa B, all those pathways we had been looking at. So in the human setting, the correlations are very high with the same pathways that we think are regulated. All right, so going back to the cytokines, we know NF-kappa B is responsible for the expression of numerous cytokines. So we did in our IA3 knockdown cytokine arrays. I'm not going to walk through all of these, but you can probably appreciate that there are lots of cytokines that are changed. And in particular, we got really interested in these two cytokines, CCL2 and CCL5. And the reason for that is that these cytokines are, number one, dramatically regulated by IA3 knockdown, but number two, known to be really important for priming for priming the metastatic niches prior to metastatic tumor cells arriving at a secondary site. So I don't know how familiar this group is with that, but essentially what's known to happen, or what's been shown to happen in some contexts, is that when you have a primary tumor, cytokines and other things are secreted that actually get to the secondary site, let's say the lung in this case, and get the lung set up for a microenvironment in which the tumor cells are going to survive better once they get there. So it's called the pre-metastatic niche. And we were very interested in this because we have these huge changes in the immune system. And so Shira Rosenbaum, a postdoc in the lab, actually did an experiment where she injected the tumor cells, either the control mammary carcinoma cells or the 2-IA knockdowns, into mice. She only let the tumors get a very small, like I forget the exact size, but she didn't let them get very large. And then she harvested the lungs and the tumor-draining lymph nodes and looked at what happens to the immune subpopulations in that tissue prior to the arrival of tumor cells. And in fact, I'm not going to show all the data, but she also did PCR and other data to show she didn't really have any tumor cells at the secondary site yet. So she harvested all the tumors at the same size. That's all that is. And then what you see here is already large changes in the immune microenvironment at the secondary site prior to the arrival of the tumor cells. So inflammatory monocytes are decreased with IA3 knockdown. Interstitial macrophages are decreased in the lungs with IA3 knockdown. And then cytotoxic natural killer cells are increased in the lungs with IA3 knockdown in the primary tumor cells prior to them arriving there. So what this suggests to us is that we're setting up the metastatic niche such that when you have IA3, it's got a microenvironment at the metastatic site that is going to be immune suppressive, so the immune cells will not recognize the tumor cells. But if you knock down IA3, the microenvironment is much more immune primed to be able to recognize the tumor cells. So here's a sort of working hypothesis of that. What we think is happening is IA3 is expressed in these cells. It induces an EMT. I don't know if this is all linked or not, but one of the things it does is affect NF-kappa B signaling. This results in the secretion of cytokines, which then brings in a number of immune populations or changes the immune populations in the lungs, and that enables the primary tumor cells to ultimately grow out. And we're now actually sort of molecularly dissecting this to show that this is actually the case. Okay, so in the last, what do I have, 10 minutes maybe? Okay, good. All right. So in the last few minutes, I'm going to tell you about what we think this all means and what we can do. So I told you that I already showed you at the very beginning that tumor cell interactions are affected by epithelial to mesenchymal transitions. Evasion of immune destruction is also known to be affected by EMT, and we've shown that the EMT molecules that we work with do the same, right? I just showed you that. And what I would say is, this is from a review that was published a few years ago, is that all of these processes that have been linked to EMT have also been linked to the molecules that I just told you about, 6 and IA, from various publications from our group and others. So we know that 6 and IA induce EMT-like processes, may not be complete EMTs, and that affects a lot of these properties. So then the question gets back to, can we target this? That was how I got into IAs in the first place, right? So some years ago, we solved the crystal structure of 6 bound to IA in collaboration with the Zhao lab. And the idea of that was that we were going to try to develop inhibitors that target the interface. We were hoping that we could actually rationally design small molecules that could inhibit these two proteins from interacting, and that would be a way that we could target tumor progression. We have done some of that, and we've actually published some of it. I'm not gonna show that today. Here we go again. Oh, and now it flipped back to the beginning. Give me 1 second. I got it. It's back, right? Okay. All right. So what I'm going to focus on again is this phosphatase activity of IA, because to me, this is probably the better way for us to do this ultimately. Okay. Oh, it stopped letting me forward again. Okay. So the reason I like the idea of targeting the phosphatase activity as opposed to the interaction is the following. So going back to MYC, I told you that IAs regulate MYC, and that has something to do with the immune effect, right? Well, it turns out many years ago, 6 and IA were shown together to regulate MYC transcription during normal embryogenesis. So it's critical MYC upregulation in response to 6 and IA, actually results in a massive proliferation that occurs during, in this case, I think it was the kidney. And so this is actually a required function of 6 and IA. Turns out that IA also affects MYC protein stability. So IA is affecting MYC on numerous levels. It's affecting transcription with 6, and it's also affecting its stability. And the way it does that is essentially MYC normally has these 2 sites that get phosphorylated. And when a phosphatase called PP2A coupled with this particular subunit dephosphorylate MYC at this residue, S62, it leads to its degradation. So what was shown in this paper by Zhu and colleagues a few years ago was that IA can actually remove this phosphate group at the T58, and that stabilizes MYC. So what we showed is it's actually not a direct, it's not an intrinsic function of IA that does it. It's actually IA switching PP2A to bind to a different subunit, which changes its specificity on MYC to stabilize it. This is a threonine phosphatase residue. But it turns out, and sorry, that results in at least the PD-L1 effects we've seen. It turns out that IA has 2 phosphatase activities. One is extrinsic through an interaction with PP2A, and the other is intrinsic as a tyrosine phosphatase. And it turns out that tyrosine phosphatase is implicated both in transcription mediated by IA, but also in the ability of its we don't know how exactly, but in the ability of it to act as a threonine phosphatase. So if we targeted the tyrosine phosphatase, we should be able to inhibit many functions of IAs, right? So that's what we wanted to do. I know we don't have a lot of time, so I'm going to try to run through this fast. But essentially what we did is we did in collaboration with the NIH Chemical Genomics Center, we did a small molecule screen trying to inhibit the tyrosine phosphatase of IA. Oh, I have never had this happen. I don't know if it's the system. It's back there, but not here. Okay, we're just going to go with it then. But anyway, what you can hopefully appreciate, I'm going to try to get it on my screen, because what you can hopefully appreciate is that the small molecules that came out of the screen all look very similar, right? They all have the same kind of structure. And they inhibit the tyrosine phosphatase of IA2. But intriguingly, so one of the problems with phosphatase inhibitors is they tend to be very nonspecific. So they inhibit a lot of phosphatases. And so the first thing we did was just ran our inhibitor against a number of other phosphatases. And what you can appreciate here is that a number of phosphatases are decreased with, are not affected by this inhibitor. But surprisingly to us, the IA3 phosphatase is also not affected and is highly conserved to IA2. So that was a big, big surprise to us. And so why is it not inhibiting all IAs? So we were actually hoping to inhibit all IAs because remember I told you that multiple IAs play a role in breast cancer. And so in collaboration with A-STAR and the Zhao lab and our lab, they actually saw this crystal structure of this small molecule bound to IA2. IA2 was the phosphatase we used in the screen. And what you might notice here is that it's actually binding behind the catalytic domain. Oh my goodness. Hold on. There we go. Back up. Huh. How do I get it to go back? It doesn't want to go back. Okay, well, I'm just going to have to skip that slide. But let's just say that it's binding behind the catalytic domain, and what it's doing is it's creating this cleft that no longer allows magnesium to bind efficiently, and when magnesium can't bind, the tyrosine phosphatase can't act. And in fact, we know precisely how that works. If you look at this, we can see the interactions of different residues with the small molecule. And what you see is this phenylalanine is really important for that interaction, and it only exists in IA2. It doesn't exist in the other IAs. And when you mutate this phenylalanine to a tyrosine, you completely remove the ability of this inhibitor to act on IA2. So we know exactly how it acts. Okay, so this causes us a problem though, right? For targeting breast cancer, because it only targets IA2. And so we started moving into other tumor types. We wanted to find tumor types that were very dependent on IA2 specifically. And I'm not going to go through all the data because I want to finish up and have time for questions, but bottom line is we recently discovered that glioblastoma, which is a brain tumor, is highly dependent on IA2. And if you put in a mutant of IA2 that doesn't have tyrosine phosphatase activity, you dramatically increase survival of these mice. And so we tested our small molecule inhibitor in the context of GBM. And what we were able to show is that it works. It's not great, but it works. And we had to do this in the flank because of blood-brain barrier issues. But interestingly, if you take the tumors out after you treat with this inhibitor, what you can see is a decrease in genes like MYC and other stem-related genes, suggesting that it is having on-target effects in tumors. So the cool thing is, and we just recently published this in collaboration with Jeremy Rich's lab, who's at Pittsburgh, but what's cool is we're also seeing efficacy in group 3 medulloblastomas, which are highly dependent on MYC and highly dependent on IA2. So we thought, we didn't even test this in breast cancer because we thought it wouldn't work in breast cancer. And then this paper was published last week by someone else that actually showed that, in fact, our tyrosine phosphatase inhibitor does work in synergy with anti-PD-1, so an immune therapy, to decrease the progression of breast cancer. So I guess we should have tested it, but I'm happy that someone else did because it's great to see that it actually does work in this context. Okay, so I'm going to end with that. So hopefully I've convinced you that, we have to think about targeting tumor progression in different ways. I'd like to think about targeting the plasticity and the EMT that occurs. It's a really hard thing to do. But even thinking about targeting some of these transcription factors that are involved in EMT and that serve as sort of central nodes for lots of signaling, I think has the potential to be a really efficacious way to inhibit tumor progression. It's difficult, but I think it's an important future direction for cancer therapy and something that we need to think about. And so just quickly, I think I acknowledge a lot of the people who did this work along the way, past lab members, current lab members, most of who I hopefully had on the slides and acknowledged along the way. This has been a really important collaboration with Ray Zhao and Lingdi Zhang, who was a postdoc in her lab for many years. And then a collaboration with Mike Lewis, who is at Baylor College of Medicine, and then immunologists at the University of Colorado, Jill and Ross, have been critical, and then many other people who are involved in the drug development ends of this. And with that, I'll thank my funding also and take any questions. I'm sorry about all the technical issues.
At the beginning, that differentiated epithelial cells, they're sensitive to chemotherapy, but T cells are not. Do you know what is the reason?
Why? So I think the biggest reason people think that is why that happens, although I don't think this is really the full explanation, is because the epithelial tumor cells tend to be more proliferative. So cells that undergo an EMT tend to be less proliferative, and so the idea is that, like, when you're migrating and invading, you're not dividing as much, and cell division is really critical for chemotherapies, for your standard chemotherapies. I have to say, though, in our hands, that is not always the case. So when you induce an EMT in a cancer cell, they don't always proliferate less well. In fact, sometimes they proliferate more well, or better. So anyway... So anyway, I don't know that that's really the whole explanation, and I think really people don't understand. That's generally the explanation that's given.
Can you hear me now?
Yep, I can hear you.
Okay.
Yep.
Yeah, so something that's interested me is why metastasis targets specific tissues. And so I was interested in this pre-metastatic niche and the evasion of immune destruction. The data you showed is only lung. Did you look at other tissues to see whether or not...
We did.
To address that?
Yep, we did. So we looked at liver. So we tried to look at the tissues where... So this tumor happens to metastasize much more frequently to the lung. So as a caveat, that is the tissue where it goes to. But actually, that might be why, what you're just saying. We did look at liver, and there are effects, but way less. So the lung was much more profound. So actually, it'd be interesting for us to take more tissues now where these cells never go, because your question is a really good one. Perhaps they go to the lung because that's the only place that they're really informing. What I don't understand, though, is that these are secreted cytokines. Why wouldn't they end up in other tissues and have the same kind of effects? I can't answer all that, but your observation, at least from the couple tissues we've looked at, is a good one. It's happening in the lungs to a much higher degree than the liver.
Just a quick follow-up question. So I'm very interested also in this pre-metastatic niche. I was wondering, does that kind of a similar scenario happens during growth? Like, is this something...
Like during normal...
Yeah, during normal growth.
I do not know the answer to that question because I don't know enough about development. That's such an interesting question, though, because you would think it could,
Yeah,
right? And especially when you're thinking
yeah.
about cells that are migrating to other areas of the embryo, that they're probably secreting things where they might be, I don't know the answer. You would probably know that better than I know. I don't know.
Never thought about that.
Thank you for that great talk. I was wondering, can you comment on the upstream effects? What's special about EMT cells that sort of activates that EA2 and 6-1 activation, and what's different? What's the initiating event?
Yeah. Okay, so I don't totally know the answer to this. I wish I did. I will say that at one point we started, and we're still working, so we're working in zebrafish models with a collaborator, Kristen Arninger at Colorado, where we're trying to compare what 6-1 does in normal development to what it does in cancer. And the reason we're using zebrafish is we can look at muscle development very clearly, and we can look at rhabdomyosarcomas, which are muscle tumors, right? Tumors that are in children. And in that context, what we showed is developmentally, 6-1 gets silenced by the presence of a microRNA called miR30A during development, right? And what we can see is that in rhabdomyosarcomas and human rhabdomyosarcomas, miR30A is actually downregulated compared to normal tissue. So it's possible that that level of control that normally exists is lost, right? But I think that's only one very simplistic idea, because I will say that we haven't been able to find that to be the case in every system we've looked at, yet 6-1 is upregulated in a lot of different tumor types, right? So in the muscle, that might be a special developmental control mechanism that's lost. I don't know about in other tissues. It's a great question. I get asked this all the time, and we've never been able to fully figure it out.
Related, well, not completely, very simple questions. Like when you look at, when you combine the EMT cells and epithelium cells, which triggers the epithelium cells to migrate more, to invade more, but then they don't change the surface markers, right? Which is very cool. But then I was wondering, during their migration or during their increased mobility, do you see any morphological change?
Not dramatic like you see with an EMT, but I would say there might be minor ones. And, you know, somebody brought this up to me recently when I gave a seminar, like we should profile the cells that are more migratory and invasive but are not undergoing an EMT, and we really should, because it would be really interesting, because it really decouples those two things that you usually think of as being coupled, right? So we need to look at that more carefully. There is some evidence in the literature. You made me think about this morning when you were talking about CDC42. Yes. There's some evidence in the literature that IAs actually can affect the CDC42 pathway and Rho and RAC, and but nobody knows how. Like, nobody knows the target. So they argued in this paper, it was published some years ago, and it wasn't our group, that it's not through 6, so it's not transcriptional, and that it has something to do with the phosphatase activity, but they don't know what the target is. So we've been trying to figure out targets. So maybe it has something to it. It has effects on the cytoskeleton that affect migration that don't classically change the EMT marker. Yeah.
Just only regularly down.
Yes. Yeah. Oh, I'm sorry. No.
Okay.
Yeah.
All right.
Thank you.